Currently, Dr. Bansal is the head of the Biomedical Data Science group established at the Göttingen and Center of Neurodegenerative Diseases, Tübingen in 2019 and funded by the Career Development Fellowship. Prior to this, he completed PhD from Max Delbrück Center of Molecular Medicine and Charité Medical Faculty, Charité might be Berlin, funded by the prestigious Marie Curie Early Stage Research Fellowship from 2012 to 15. And after that, broadened and deepened expertise in a number of postdoctoral positions at Max Planck Institute, Experimental Medicine, Göttingen, and the Center for Molecular Neurobiology, Hamburg. He had been curious and investigating biological questions independently using high throughput sequencing data and computational models. His group is interested in producing, integrating, and discovering patterns in large multi-omics datasets to extend the understanding of transcriptional and post-transcriptional regulatory mechanism. Integration of different sequencing data types provides an opportunity to investigate biological pathways at multiple layers like genotypes, chromatin, and transcript levels. He together with the collaborators uses iPSCs, human tissues, and mouse model to generate cell types, specific multi-omics data, and to better understand the diseases related to the brain, nervous system, such as Alzheimer's disease, frontotemporal dementia, and the Parkinson's disease. So it's a wide open to you. I feel there are lots of work you have done, and you are going to talk about the Parkinson's disease. And please keep in mind your time. Thank you so much, Dr. Bansal.
Thank you, sir, for the nice introduction. And I would also like to thank the organizers, especially Professor Chaube, for the invitation, and also I would congratulate the organizing team and his team for the wonderful effort they are doing. And it's a great pleasure to be here, and I am glad to share some of our initial results from the multi-omics data we have produced in the consortium FOUNDIN-PD, that is Foundational Data Initiative for Parkinson's disease. So before I start, I would like to acknowledge the FOUNDIN-PD consortium. As you can see, this is a huge team effort. We have Henry's and Mark's team from NIH, Peter's and my group from DZNE Tübingen, Kendall's group at DZNE, and Steve's group at Glasgow Institute, David's group at Charité Medical Center for USC, and the funding is provided by Michael J. Fox Foundation. So this is the foundation started by Michael J. Fox himself. Maybe you know him from some movies. That he was also diagnosed with Parkinson's disease in late 90s. So they provide funding especially for Parkinson's disease. So this is a next slide, but what I would like to highlight a few points from here, that in Parkinson's disease we have loss of a specific cell type. These are dopaminergic neurons from substantia nigra part of our brain, and these are the symptoms what we have, including rigidity, tremor, and balance abnormalities. And there are more than 10 million people worldwide, and also the last calculations what we have in India was more than 500,000 PD patients we have that was calculated in 2016. But this number is expected to shoot up as we will have an aging population in coming 2 or 3 decades. And the incidence of PD increases with age, as we already know. And to give you some perspective of how much money is spent on the treatment, this is around $52 billion in US alone. So we can see that even in India, when we have a huge aging population, how much care and how much money we have to spend for this particular disease. So here in this image, I try to summarize the genetic architecture of Parkinson's disease. So what you see here in x-axis is the frequency of population, and y-axis the risk of PD. So what you see here at the top are the causal or the known genes which are causal for PD. This includes C-nuclein, that is C and S and CA. This is one of the first genes that has been associated with Parkinson's disease. Then we have strong risk factors like GBA and LARC2, and the factors which are identified by GWAS. These are more common and frequent in population. And with latest GWAS, we already have around 90 loci or 90 genes which has been associated with Parkinson's disease. And this number is expected to go beyond 90 when we have a large or relatively larger cohort of PD patients. So we have many gene candidates here, but what deficiency or how there is a loss of dopaminergic neurons in substantia nigra of human brain. So we can ask very precise questions that the functional characterization of these mechanisms, what are the pathways that are connected by genetic interactions, what is the chromatin status of these loci, transcription and translational changes, and many more. So to answer all these questions, what we would need is a kind of multi-omics approach, meaning generating data at different levels and integrating it to see what pathways are getting perturbed in Parkinson's disease. So for this, we are using iPSCs as our model. As you know, that what we can do with iPSCs are to take or to reprogram the cells from patients, and then because they carry the mutations which are relevant to PD, we can differentiate them to a particular cell type, in this case dopaminergic neurons. So why this is also important for why we are using iPSCs in many countries, including Australia, Singapore, China, and even in US now. In 2021, they got a green light to use this iPSC or embryonic stem cell drive dopaminergic neurons as a kind of therapy, so that you can use them directly in substantia nigra of human brain. So to put this part in perspective, in terms of personalized medicine, we start with a patient, for example, here PD patient, or as Dandapani also showed with or a cardiomyopathy. We reprogram these cells to iPSCs, and then we can also correct these iPSC lines for the mutation, for example, GB or LAR2. And then these corrected iPSC lines can be differentiated to a particular cell type, in this case dopaminergic neurons. So here, before transplantation, what is very important is to check the quality of these cells. So here, this is what we are doing with the quality checks at molecular level using the multi-omics sequencing data. So we start with around 95 iPSC lines, and we have these iPSCs from PPME initiated, which have the iPSCs from health controls, patient IPD, and the patients which contain PD relevant mutations, that is GBA, LAR2, and PD. So what we do is we differentiate these cells to dopaminergic neurons and collect these cells at different time points to generate multi-omics data at different times. So we have genetic, where we do genotyping, mitochondria DNA-seq, short and long read whole genome sequencing. At regulation, we have DNA methylation and small RNA-seq, and the chromatin level ATAC, both bulk as well as single cell ATAC-seq. And at expression, we have RNA-seq as well as single cell RNA-seq, and some of these samples were also underwent for long read RNA sequencing. So what I am going to do now is to present some results from single cell RNA sequencing data. And before that, just a quick introduction, what we are using as 10x genomics. I think many of you are already familiar with this. So we'll not go into technical details, but the idea here is to capture the single cells in oil droplets along with the beads which contain a cell barcode. So what's going to happen when we do the pair-end sequencing is that we have read 2 which contain your transcript, which will be mapped back to the genome, and then we have a cell barcode which will keep the track of this transcript from which cell this is coming from. So our first aim is to create a gene cell expression matrix, which is basically that in columns you have number of cells. This can go beyond thousands and even millions nowadays. And then we have genes in the rows, and the numbers represent how strongly this gene is expressed in a particular cell. So this is just a numeric matrix, and you can apply, or you can think of many computational or statistical approaches to apply on this numerical matrix. So just to give you an overview, there are many methods to analyze this single cell data, but I'm just showing you a glimpse of some of the tools we are using. So for starting with the matrix, we can have a cell level analysis, which is starting with the clustering. So we cluster the cells which are showing the particular pattern of gene expression, and then we visualize them, for example, using UMAP plot or DZNE plot. And then we also have at gene level, we have expression of these markers in a particular cell type. So meaning we can also identify new markers. And then we can also do pseudotime analysis, meaning that if there is a gene that is kind of following a biological pathway in your particular data set. And then we can also do gene-gene correlation, as you know that genes are not working alone, but in combination with another genes. So this is what we have now, the single cell data from iPSC-derived dopaminergic neurons from our FOUNDIN-PD project. So what you see here on the left side is a UMAP representation of around 400,000 cells coming from 83 samples, and each dot here represents a single cell. So on the right side, so basically what we see here is the two big clouds of clusters. On right sides, we have mainly the progenitor cells, and on left sides, these are the ones which we are producing. These are dopaminergic neurons. Further are divided into two, relatively mature and relatively immature, and this is supported by our pseudotime analysis. As you can see here, the pseudotime is higher in the golden or relatively mature dopaminergic neurons. So we also use the known markers, for example, TH, this is the known marker to define our dopaminergic neurons, and many other markers which are known to be expressed particularly in progenitor cells. So this is just to show you the quality of the data. So one of the first thing what we wanted to check is, as these cells are produced in the lab, how comparable these are with the adult human substantia nigra dopaminergic neurons. So luckily what we had is the publication from Weber lab, where they have produced the single cell data from adult human substantia nigra. And here you can see that the type of cells, we have oligodendrocyte, astrocytes, precursor cells, as well as PH neurons, which are our dopaminergic neurons. And when we do the correlation with the Weber data, with the FOUNDIN, the single cell RNA data, we were very glad to see that we have a highest and significant correlation with the PH neurons with our relatively dopaminergic neurons. So to conclude this part, it tells us, although not perfect, but we have cells which looks very similar to dopaminergic neurons of adult human substantia nigra. So the next question was that, are these cells relevant for Parkinson's disease? So for this, what we did is to integrate our single cell data with the PD jivas. So basically what we are seeing here on the right side is the enrichment of PD relevant SNPs in a different cell types what we have produced in FOUNDIN-PD. So as expected, what you can see here is that the dopaminergic neurons, both the populations are significantly associated with PD jiva SNPs. So this tells us that the cell types what we are creating are indeed, they are useful for modeling the PD related genetic risk. So looking at the cell type composition sample wise, so here what you see is each bar represents a sample, and then what is the cell type composition of that particular sample. So first thing you will notice here is that we have a lot of variation here, for example, in dopaminergic neurons, but we don't find any pattern or any significant difference between the cell type composition of the groups. For example, if we compare dopaminergic neuron clusters between healthy control or GBA positive cohort. And the second thing you would notice here is that as we are producing dopaminergic neurons here, still we are getting a very low percentage of these neurons. So for example, only on average we get 20%. So using this data set, what we can ask is that are there any some patterns which we can learn from this multi-omics data to make this differentiation protocol more efficient? Because if we would like to use these cells for the transplantation, it's very important to have a higher efficiency of this differentiation protocol. So I'm going to show you two examples. One is at the gene level, and the second is at the gene-gene correlation level. So for the first thing, what we did is to use the bulk RNA-seq data from FOUNDIN-PD. And here we just show that we have a day 0 where the pluripotency markers are going down, and the markers which are in dopaminergic neurons are going up from a MEP2 and TH. So what we did is to use the expression of these genes at day 0 and correlate it with differentiation efficiency at day 65. So basically what we are asking is, are there some genes at the iPSC levels which can already predict the differentiation efficiency at day 65? So we found these 5 genes which we think if we sort the cells initially at iPSC level, hoping for to get more percentage of dopaminergic neurons. So as a second example, what we are doing here is the gene-gene correlation or gene modules. So for this, we are using a published tool, scenic. Basically, this is a 3-step approach. What we are doing is to find first the gene combinations or the genes which are expressed together using a random forest approach. And in the second step, we use the biological information, for example, the enrichment of motifs in this particular gene module. So then we start calling them as regulons. And in the third step, we ask in which cell types this particular regulon is active. So using this approach, in both single cell RNA and single cell ATAC-seq data, we found that these particular transcription factors, one cut, which is one cut 2 as well as one cut 3, they are specifically expressed in dopaminergic neurons. And these are gene modules. Just to confirm here, you can see in dopaminergic neurons, you see a higher or relatively or a specific expression of these genes in dopaminergic neurons. So what we are doing now is to stimulate these transcription factors alone or in combination in hoping for to get more percentage of dopaminergic neurons at day 65. So there are many other things what we are doing. In FOUNDIN-PD, we have eQQL analysis, epigenetics in FOUNDIN-PD, and then we are also collaborating with Manu's lab for non-coding sequences and long read. And our ultimate goal is to analyze and integrate this data, the multi-omics data, to find patterns which are specific for PD cohort as compared to relative to healthy population. So to summarize, what we have done so far is successfully differentiated these iPSCs to dopaminergic neurons, and these are very useful, as I have shown you that these cells can be used in context of genetic risk, and the data is freely available at PPMI and our FOUNDIN-PD browser. So with this, I would like to thank you and happy to take your questions.
Thank you so much. And since you are coming from working in Germany, so thank you. Thank you. So this is a very nice work. So just I can take 1 or 2 questions. Before that, I just have a kind of observation. So what about the sexual dimorphism? I mean, the Parkinson disease in the males and females. Did you get any kind of? Okay. I feel that there will be some kind of differences. So I feel that it may be your future. Research about this sexual dimorphism, because there's a muscle weakness and all kind of things and some kind of because I'm coming from anthropology, so there are some anthropometric tests like the hand grip strength in case of Parkinson, and also body water, extracellular body water and intercellular body water can be measured. And this case there might be some case of sexual dimorphism. So I'm looking forward to see that whether in case of the differences in the sex. Thank you so much. So we can take 3. Okay, just three quick observations. Thank you so much. You can start on.
That's nice word. I just not ask a question, but let me say that we are also working on Parkinson's disease and Parimantas Center for Genetic Disorders. What is there? And we are also associated with Michael J. Fox Foundations having things are going fine so far. But we need some acceleration of these, especially for preparing iPSC cell line, which I'm trying having from here in collaboration in Bangalore and other places, where you could be of some help to us. That's all my question.
Sorry.
Can you help us in getting this iPSC cell made?
Definitely. So the procedure is for the PPMI database, we can always apply for this iPSCs. So we have to kind of do some material transfer. But if you are asking to generate this in the lab, so you want to reprogram in the lab?
Yes.
Yeah, so these are well established protocols. We can definitely do that.
I'll talk later.
Yes.
Wonderful talk, Vikas. I was just wondering that your entire work was on actually dopaminergic neurons, but all the genes that you have initially showed, how do they really respect dopaminergic neurons? Because none of them actually are related to dopaminergic neurons.
Great question. So two of them, which are present as CPPA and GP, they are in neurons. They are expressed. But LAR2 that is expressing in oligodendrocytes. So what we are doing now is we are applying for funding to get these cells to differentiate to oligodendrocytes. Because I really think that if we only consider neurons in this kind of diseases, we are not going to learn much because oligodendrocytes and microglia are equally important. So what I am aiming for is to create a data, or at least this multi-omics data from different cell types. So we take the same iPSC cells, differentiate them to neurons, oligodendrocytes, microglia, and then we have the complete picture.
Yeah, in fact, in communication, that there's very interesting paper that recently came out about the oligodendrocyte methylation evolution pattern, oligodendrocyte methylation pattern from the evolutionary perspective, with respect to CGL methylation and CH methylation. Then, it is a very, I think it came just last week in the
Related to Parkinson's disease?
No, general population from the evolutionary perspective.
Excellent presentation. In terms of integrated approaches, have you got any plans of including that microbiome? Have you got any plans?
Wonderful idea, but right now we have no funds to do this. But there are other labs who are doing this, also using the same patient lines. So what they are doing, because this database, it has different clinical features of a particular individual. So this is not what we are doing. We are working at iPSC levels. So they are also integrating other clinical information. But I think that's going to be really useful to integrate that microbiome.
I hope this is a time that as we successfully ended up the whole discourses and all the lectures. It was wonderful. So I feel that summarization from the chairperson will be going.
Thank you, sir, for the wonderful session, and I would request Dr. Mishra to please facilitate Dr. Bansal, and I request Priya to please accompany sir. Now I would like to request our session chair to please summarize the session. Please, sir.
Okay.
And then we are thankful for all the speakers for the insightful talks in session 5, which witnessed complex molecular DNA research to reveal the basis of the genetic basis of the any kind of diseases. And for the first speaker, Professor K. Thangraj, who discussed the Indian population size even before regional clinical setups in infertility period, and different kind of cases which is affected, mostly affected by genetic cause, like the infertility rate relation with child mortality, education rate, palindromic, and is very interesting research and infertility, mitochondrial understanding for the relatives and all this making correlation. It's very interesting, and this research has lots of clinical relevance which can be further translated into some kind of effective tool. So this type of talk is very, very informative. And then we have witnessed Dr. DZNE talk, which mostly focus on ancestral diversity. which has used lots of genomics approaches and specific adaptive traits, which give lots of insights about the evolutionary genetic history. He showed lots of evocation sites and lots of prehistoric sites which can be correlated to ancient DNA research. So this is very, very interesting to see and correlate with our ancestral, or you can say historical heritage with this field. And lots of challenges also he mentioned, like 1% success. It means lots of inputs required in terms of resources and everything, expertise, and more advanced tools to address all these challenges. 1% success, it means very, very challenging field and DNA. And we can correlate lots of population migrations, and we work on Rakhigarhi samples, Kashmir region, Neolithic and Megalithic, Northeast region, Nagaland, Mizoram. So almost the whole India he covered his research. And one of the most important things which I observe is relation with the clinical relevance with the pathogen like mycobacterium leprae. So this kind of any kind of virus bacteria which can be infected in our older population. So understanding the mechanism and other there really has a lots of potential to understand the clinical phenotypes. And then we have Dr. Dandapani Perudursukai from Eastern Bangalore. He is mostly discuss about the gene discovery and mechanism and therapeutic advances, and in which he discuss about the pluripotent stem cell and drosophila research, and in therapeutic advances he discuss about the mevacatum drug activity. So his research is also very informative for the cardiomyopathy disease and understanding its complex mechanism. He also his research is mostly has a translational potential, which is the most important part of this organization also. And then we have a Professor Manjunath Joshi talk, which is immunogenetic research on neutrophil extracellular traps and nets. So nets involved in several pathogenesis mechanism of various diseases, and he emphasized mostly on type 2 diabetes, their functional heterogeneity, and then its correlation with vascular disease in stroke, and we work on homeostasis. So this research is very, very important for lifestyle diseases, particularly any kind of cardiac disease, any other fatigue disease. So again,